Okay. Wait, why is this? That was weird. Why is it? Okay. بسم الله الرحمن الرحيم والصلاة والسلام على أشرف الأنبياء والمرسلين نبينا محمد وعلى آله وصحبه أجمعين. Assalamu alaikum everyone. Alhomam Dabaliz here, and I'll be doing this PAL session about the cell signaling 1 through 3. So we'll go through them one by one. We might have a break like in the middle, but inshallah it won't be that difficult or that much. I can't open the camera unfortunately, but inshallah that won't be a problem. So our first thing we'll be talking about mostly is the G protein coupled receptors. This is the bulk of the three lectures, the G protein coupled receptors. The rest should be easier, inshallah. Even this one is easy. They're all simple. They just need you to have like a good imagination and just follow the steps as much as possible. If anyone has any questions, they can always ask whenever they want. I'll answer whenever I feel like it's appropriate for me to answer. And you just use the chat mostly. If you want to open your mic, that's fine as well, but because it's being recorded, you might not want that. And it's a personal preference. So starting with the first slide is just talking about the different types of cell communication. And I think most of you already took this like in GI and different blocks. So there's the direct contact through gap junctions, for example, from cell to cell. There is the ones that are paracrine, so it's in the same organ or like right around itself. It just like releases the signals and immediately takes it. And then there's the other ones like the endocrine, which sends it to the bloodstream, and then the synaptic, which is through neurons. That's the main ways that cells can communicate with each other, right? Okay. So now the idea of signaling, there's a few terminologies that we should all have in our minds when we're talking about signaling. One of the terminologies we're talking about is what is signaling. It is the communication between cells. Simple as that. Then we have what's the signal transduction, the ligand, and the receptor. Starting from the raise my voice. Give me a second. I think my voice is as loud as I can go. Okay. So starting with the ligand itself is just the signaling molecule. So maybe we're talking about a hormone, a steroid, a protein, anything that will bind to the receptor, basically. Okay, it can even be we had like in MSK you studied ligands like acetyl-CoA, and different ligands basically. All of these, acetylcholine, I mean, all of these can act as ligands. Then we have the receptors, which are the molecules that will bind to the ligand and start the basically the cascade or the signal transduction. And lastly, the signal transduction is just the entire process where the signal from outside, which is the ligand, is translated as an action inside the cell. This is what a signal transduction is. Okay, so it is the whole process of turning a signal from the outside, which is our ligand, into a response in the inside, which is everything we'll be talking about right now. These responses can be a plethora of things, like over here they mentioned a few of them, like altering the metabolisms, like for example activating gluconeogenesis while stopping glycolysis, for example. Altered gene expression, which is all the transcription factor stuff we'll be talking about today, and altered cell shape or movements, which you took a bit of in like endocytosis and elongation of smooth muscles, etc., and epithelial cells. So the components we have is ligand, receptors, and the intracellular signaling proteins. These intracellular signaling proteins can be enzymes, messengers, different types of proteins, and we'll be talking about a lot of them right now. Okay. So now here they're just mentioning a bit about the receptors. Some receptors are found on the membrane. Some receptors are found inside of the cells or inside of the nuclei, and we will be talking about these much more. Now, the thing that you need to know is that membrane receptors have a lot of signal amplification and not just signal transduction. So just one ligand binding to one receptor can cause a lot of or like it can cause a signal to amplify exponentially, like so not just one pathway. No, it will go into like 100 of the same thing. So for example, a receptor that is causing the formation of a cyclic AMP will not just form one cyclic AMP, it will form 100, 1,000, a million of these cyclic AMPs. And all of these can go and cause a response to happen. On the other hand, when you're talking about nuclear receptors, it is a one-to-one. So a steroid will bind to its own receptor, one receptor, and this receptor will go to the DNA that it wants to go to. Because you don't have like a million of the same copy of your DNA, you only have one copy of your DNA, two copies maximum of your DNA, right? So if you just have one or two copies, you don't need to make like 100 steroid receptors. You just need one to act on your one DNA strand, and that's it. Okay? So that's one thing that you should know. And membrane receptors are associated with signal amplification. Nuclear receptors do not need the amplification. Okay? They are already, there's like a high concentration of them in the cell itself. Okay? The second thing is that membrane receptors are associated with hydrophilic ligands, because the hydrophilic ligands cannot enter inside the cytoplasm, so they need to work on the membrane, while the nuclear receptors, which are already present usually inside of the cytoplasm or in the nucleus itself, these ones are associated with hydrophobic ligands like steroids, vitamin D, A, E, K, all of these. The hydrophobic stuff that can go through the membrane. Okay. So again, the signaling molecules, our ligands, can come in many shapes and shapes. We have steroids, we have amino acid derivatives like acetylcholine, epinephrine, dopamine. We have eicosanoids, which are our not interleukins, our prostaglandins and our interleukins and prostaglandins. Interleukins are our prostaglandins here, and our polypeptides, like for example insulin, that's a full hormone. So these are the different forms. Each one of them can be an extracellular signaling molecule, a ligand. Okay. Now, this is for the ligands. Now, talking about the receptors. The receptors can also come in many shapes and sizes. The receptors can be a G protein coupled receptor. That's what we will be talking about mostly right now. We have receptors that are associated with the tyrosine kinase. They have the ability to be tyrosine kinases, ligand gated channels, steroid hormone receptors, and all of them, again, we'll be talking about them right now. Okay. So, and this is just a picture to visualize all of them. Again, we'll have a lot of visualization through the lecture, so no need to look at them. And now let's focus on our first and main subject, which is the G protein coupled receptors, or guanosine protein coupled signal transduction, whatever. Okay, so these receptors are, as you can see here, they are like helical, as you can see here, 6 transmembrane receptors. Our N terminal is over here, and our C terminal is over here. The N terminal will be binding to the ligand. The C terminal will be doing the magic inside of the cell. Okay. They are the most common type of receptors in the body. You will notice that most of your adrenergic receptors, the alpha and beta, both of them are G proteins. A lot of the acetylcholine one are also G proteins. So you can imagine how much of the G protein coupled receptors we have. The glucagon is a G protein. The epinephrine, the norepinephrine, the acetylcholine, all of these, I believe, all of them are G proteins or can be G proteins. So you can imagine how much we use this form of receptors in the cells. Okay, so again, N terminal, this binds to the ligand. C terminal does the magic inside, and then you have 1, 2, 3, 4, 5, 6, 7 transmembrane domains that are stabilized via disulfide bridges. Okay, they are only found in eukaryotes, and the ligands that can bind into them are can be anything. It can be light, just light, like your retina has G protein coupled receptors that activate through lights. Odors, pheromones, hormones, neurotransmitters, all of these can work through G protein coupled receptors, and all of these can be ligands for this receptor. Okay. Here we start talking about the structure of it all, and the main thing you want to look at is the G protein itself. This is the receptor, and the receptor, as you can see here, again, the N terminal dealing with the ligands, as you can see here, and then the C terminal dealing with the inside. Specifically, the C terminal is dealing with what? With the G protein. The G protein is a it's made of multiple parts. It's a heterotrimeric, as you can see here. So it's made of multiple parts. These parts are called the alpha and the beta and the gamma. Okay. The main active part is the alpha. Okay. This is the one that acts on anything. This is the one that will do the action. The beta and the gamma are just there to keep the alpha not active, inactive. When the beta and the gamma are bound to the alpha is not active. When the alpha is unbound to them, it can do whatever it needs to do. Okay. So the active part of the G protein is the alpha subunit. All right. Now, when is this alpha subunit actually active? Well, back to the name, it is called a G protein. Why is it called a G protein? It's because it has the guanosine, which is the GDP. Okay. When this GDP is exchanged for a GTP, so it gets phosphorylated or has like 3 phosphates, this alpha subunit can dissociate from the beta and the gamma and becomes active and works through the pathway that it should work through. Okay. So the question is, when is our alpha subunit active? When it is bound to a GTP. When the GTP is exchanged for a GTP. Is this clear? Okay. All right. Just take off the direct messages. Okay. Now this is our first part. Now, talking about this whole GTP, GDP stuff, we have how do we get the GDP and how do we get or how do we go to GTP and vice versa? We have something called a GEF, which is a guanosine nucleotide exchange factor. Okay, so a guanosine exchange factor. What does that mean? It means it exchanges the guanosine, which is the GTP or the GDP, between each other. The GEF is capable of exchanging a GDP for a GTP and vice versa. Okay, so the GEF can activate the G protein, the G alpha subunit, or it can inhibit the subunit. Okay, that's the first part. The second one is the GAP. This is a GTPase activating protein. Where is this GTPase coming from? And what's the GTPase? A GTPase is an enzyme that can turn GTP into GDP. So it dephosphorylates the GTP, correct? Okay, so where does it come from? Well, every G protein, like the alpha subunit itself, has GTPase activity, okay? So the alpha subunit has GTPase activity. The gap basically just activates this GTPase activity, which leads to the cleavage of ATP in GTP into GTP, making the alpha subunit inactive. So what are our ways of inactivating the alpha subunit? We have 2 ways: either via the GEF reversing exchanging a GTP into GDP, or by the GTPase activating protein activating the GTPase of the alpha subunit and cleaving the GTP into GTP. So we have 2 methods. Is this clear? Oh, my back. Okay. So this is for this part. Okay, so here again, it's just an overview of what happens. So we have binding of the hormone, activated G protein complex, the active G protein complexes, the G protein, the active one, activates other enzymes, and these other enzymes will start the signal transduction pathway. That's it. So now we will be talking mostly about what are these other enzymes. So we know what the G protein coupled receptor does. We know what the G protein is, right? We know these things. Now let's talk about downstream into here. So talking about downstream into here, you have the G protein coupled receptors, and here we start talking about the enzymes. So first of all, here is just mentioning what are the components. Transmembrane receptor, we mentioned that. G protein trimeric complexes, we mentioned that. Next, the enzymes. Now, in general, okay, the G protein can activate many types of enzymes. So many types of enzymes. But we will focus on three kinds of enzymes that the G proteins will play with. Okay, we have the Adeline Cyclase, the Phospholipase C, and the Guanine Cyclase. We have these three enzymes. We want to focus on them as much as possible. The rest, you might take them through your years. It's fine. But right now, we will be talking about these three. So the Adenine Cyclase, or Adenyl Cyclase, is the one that will form cyclic AMP as its secondary messenger. So it's a very important thing to always know what the secondary messenger is. The primary messenger is your ligand. Okay, so it's epinephrine, insulin, I don't know, acetylcholine. All of these are your primary messengers. Your secondary messenger is the messenger that is working inside of the cell, that is going from your receptor to the place that it should be working. So we're talking here about the cyclic AMP in case of Adenine Cyclase. We're talking about the IP3 and the DAG in case of what? In case of the phospholipase C. Okay? And we're talking about the cyclic GMP in case of the Guanine Cyclase. All right? Now there are so many forms of the alpha subunit, but mostly we will want you to know about the I, inhibitory, S, stimulatory, Q, this is the one that has that deals with the phospholipase C, and the 1213, which is the one that deals with the Guanine Cyclase. Okay, now we'll take them one by one, I think, right now. Yeah. So here again, it's just a table that's showing you each one of them, what activates it, and what does it activate. So the stimulatory, this one, it will activate your Adenine Cyclase, so it will give you cyclic AMP. Okay, and what activates it? Beta adrenergic amine, so beta agonists, glucagon, PTH, thyrotropin, so TSH, CTH, all of these can activate, work through the Adenine Cyclase. And then you have the inhibitory one, which basically is the exact opposite. This one stimulates Adenine Cyclase, this one inhibits it. This is your parasympathetic system, your alpha adrenergic, and many other neurotransmitters. And then you have this one that works through the phospholipase C, and this is the one that you can find it actually in your eyes, in your retina. Photons activated. Okay. Okay. So now, I think I already mentioned all of this. The transformation of GTP to HADA. Yeah, I already mentioned all of this. Okay, we're talking about the Adenine Cyclase here. Does anybody have any questions until now? Oh, okay, I'll keep going then. Bismillah. So, all right, now we're talking about one by one of the G proteins, and we're starting with our Adenine Cyclase. This one is very, very simple, as you can see here. It's just showing you in all. You have a hormone that binds to your receptor. This leads to exchange of your GDP to a GTP. Alpha subunit is activated. It goes to your Adenine Cyclase. It activates it, and this turns your ATP into cyclic AMP. And the cyclic AMP will do stuff. Basically, it will do its receptor, its messenger stuff later, which we will talk about in just a second. Okay. Yeah. Where is the good slide to talk about this? Okay, so we start with here. This is our Adenine Cyclase. Okay. Again, GTP, alpha subunit, activates Adenine Cyclase, ATP turns to cyclic AMP. Now, the exchange is through GE. It's only through GEF. So GDP turning becoming GTP is GEF. But turning from GTP into a GDP can either be GEF or GTPase. Both of them can work, can inactivate. GEF is the only one that can activate. Okay. Now, talking about cyclic AMP, what does the cyclic AMP mainly work on? It works mainly in most parts of your body or a cell. It will activate this protein kinase A. A stands for adenosine. So protein kinase A is now activated, and this one will phosphorylate many proteins in your cells, and this will lead to many responses. Okay. I don't know if you remember, but for example, if you remember in gluconeogenesis and glycolysis, in glycolysis, all the enzymes are activated when they are unphosphorylated, while in gluconeogenesis, they are activated when they are phosphorylated. This is what happens. The glucagon, which is the one that activates gluconeogenesis, goes through exactly this. It is a ligand from GPCR-GS, which activates Adenine Cyclase, cyclic AMP, pKa, and then phosphorylase the enzyme inside of the cell, and this leads to activation of gluconeogenesis. Okay, so that's like an example of where does this happen. Epinephrine, glucagon, all of these work through this reaction of turning on protein kinase A, which will phosphorylate proteins, which will lead to your cellular response, whatever it is. Okay. This is just, again, this is showing you the same thing, but now it's telling you how do we turn off this mechanism. Yes, we are making cyclic AMP, and we are activating our protein kinases, but that's how do we turn this off? We turn this off via phosphodiesterase. Phosphodiesterase is capable of converting cyclic AMP into just normal AMP. Okay, so in general, phosphodiesterases work to turn cyclic nucleotides into normal nucleotides. So from cyclic AMP or cyclic GMP into normal AMP and normal GMP. And this is how we can turn off the signal. So we turn off the signal from the receptor itself. So from here, we will turn it off from here by stopping the ligand, and turn it off from here by destroying our secondary messenger. And this is how we stop the system here. Okay. So how do we regulate our cyclic AMP levels in the cell? By decreasing or activating and inactivating the Adenine Cyclase, and by producing phosphodiesterase. That's it. So we have two mechanisms. And that's it for the Adenine Cyclase. Is this clear? Actually, yeah. Okay. Is this clear? Okay. Now I'll skip through the phospholipase C just a second to go for what happens if our Adenine Cyclase is damaged. Okay. Now we have two main examples to talk about to mention this, how the activity can be increased in Adenine Cyclase receptors. Okay. And these are the Cholera Toxin and the Pertussis Toxin. The cholera, I think everyone knows what cholera is. It's the very, very heavy watery diarrhea that's like, it's just water. There's no stool there. It's just water, and it's caused by bacteria. And then there's the pertussis, which is the whooping cough associated dicky, which we get vaccinated for by Bordetella pertussis. Okay? These two work on dysregulating the Adenine Cyclase receptors, and that leads to different manifestations depending on the cells they affect. So starting with, this is our cholera toxin. Cholera, where does it happen? It happens in your intestines, in your enterocytes, okay? In your intestinal cells. Cholera toxin can enter through the cell, and it will inhibit something. What does cholera toxin inhibit? It inhibits the GTPase activity. Okay? So the GTPase activity of the G protein is inactivated by the cholera toxin. This means that the GS is always activated. It is always activated. This means you will always get more and more cyclic AMP. You will have a lot of cyclic AMP. In the enterocytes, what the cyclic AMP does, it turns on pKa, which will phosphorylate the CFTR, if you remember these from GIT. The CFTR channel, the cystic fibrosis transduction receptor, I think, will push chloride out of the cell, and water follows the ions. And that's why we will have a ton of water outside our cells, and there goes the diarrhea. Okay? So cholera, how does cholera works? Cholera works by turning off the GTPase. So you will have a lot of activity of the GS, a lot of cyclic AMP, a lot of activation of the CFTR, a lot of chloride and water going out, and diarrhea. That is cholera toxin. Okay? Now compare that with pertussis toxin. The pertussis, on the other hand, works on a GI. Now, what is GI, if you remember? What is GI? We said GS stimulates Adenine Cyclase, GI inhibits Adenine Cyclase. Pertussis stops the GI. Okay, so pertussis inhibits the inhibitor, right? So if we inhibit the inhibitor, what happens? What happens is we will have constant activation of the Adenine Cyclase. Adenocyclase, again, it will be constantly activated, a lot of cyclic AMP, and this will lead to the problems that happen with cholera, with the pertussis, like the specific cough that happens with pertussis because of the ion mishaps and the water in the lungs and stuff like that, or in the trachea and stuff like that. Okay? So is this clear how pertussis works and how cholera works? And this is just a slide that shows comparing them between each other. This is the cholera toxin. This is the pertussis toxin. This one stops the inhibitor from working. This one stops the GTPase from working. That's it. Okay. These slides, by the way, are taken from Dr. Ahmed himself. He made them like 4 years ago. I don't know if he gave you these slides or not. But these are like the only slides. He made these slides and just just a few images, and that's it. Okay. Now that's it for our Adeline Cyclase. Let's talk about the other pathway, which is the Phospholipase C pathway. The Phospholipase C pathway is a very important one, especially in muscles, as it is one of the main ways that we get the calcium outside of the sarcoplasmic and the endoplasmic reticulum, leading to the muscle contraction and stuff like that. Okay. So this is one of these things that you kind of already took before, but now we're taking them in a different way. Okay. So here it's the same thing, just like we were talking here, Adenine Cyclase. So it's the same thing. We get the hormone, we activate our alpha subunit, we make it GTP, we activate it, and that will go to the phospholipase C, which will activate it. Now, what type of G protein is this? This is the Q. The Q works with phospholipase C. The S and I work with Adeline Cyclase. Okay, just remember that. Now, the GQ, the activated one, the alpha subunit, will go to phospholipase C and activate it. Now, what does phospholipase C do? It is a lipase, so it breaks lipids. The main place where you will find lipids in the cell is in the membrane. So this, or phospholipids specifically in the cell, is in the membrane. So it will go to the membrane and just break one of the one of these PIP2, the PIP2, the bisphosphates, install bisphosphate, whatever. It will break it into an IP3 and a DAG. Okay, the DAG is the part that will stay connected to the membrane. The IP3 will become free flowing in the cytoplasm. Okay, and each one of these is a secondary messenger, not just IP3. The DAG and the IP3 is a secondary messenger. The IP3 will go to the endoplasmic reticulum, bind to a specific receptor, and this leads to calcium being shot out of the endoplasmic reticulum into the cytoplasm. The DAG and the calcium together will activate protein kinase C, and the protein kinase C will phosphorylate proteins, which will lead to different effects in the body. Okay, so the main points you need to know is how to compare between the phospholipase C pathway and the Adenine Cyclase pathway. Okay. The Adenine Cyclase pathway, I'll be calling it AC pathway from now on, just because my tongue hurts. In the AC pathway, your secondary messenger is what? Is cyclic AMP. Okay. Cyclic AMP activates what? the protein kinase A. Okay, that's it. On the other hand, in the phospholipase C pathway, your secondary messengers are the IP3, the DAG, and the calcium. All of these are considered your secondary messengers. The IP3, the DAG, and the calcium. And what's the protein they activate? They activate protein kinase C. Okay, that is your comparison between the two. Phospholipase C, the secondary messengers are DAG, IP3, calcium. The protein activated is protein kinase C. In AC pathway, it is cyclic AMP, that's the secondary messenger. The protein activated is protein kinase A. Is this clear? Point 5. Yeah, so 0.5, phospholipase C as an enzyme, what does it do? It breaks one of the phospholipids that are found in the cell membrane into DAG and IP3. It breaks them into these two. IP3 goes to the endoplasmic reticulum, shooting out the calcium. DAG goes to the protein kinase C and HADA. As you can see here, this is the cleavage. So this is the PIP2. It is cleaved like this. So you have the DAG and the IP3. And as you can see, because this is the part which becomes the DAG, it stays bound to the plasma membrane. This is the part that is not bound to the plasma membrane. So it's the IP3, which is free flowing in the cell. Sorry, my voice was far away. I had to dodge something. Okay. All right. So this is okay. Wait, what was this? Yeah, the GTP. This is the Pertussis Toxin. Here it's just showing you the different families again. And here we're talking a bit about the Cyclic Gmp. Now, Cyclic Gmp is another secondary messenger. Just like Cyclic AMP, we have Cyclic Gmp. Cyclic Gmp is mainly associated with interactions that concern nitric oxide. Okay, nitric oxide is generally something is a substance that will cause vasodilation. You need to remember that it causes vasodilation, mostly the veins. Venodilation mostly, okay? And this is just showing you one way of it happening. So nerve cells will activate endothelial cells nitric oxide synthase, which will turn arginine into nitric oxide. The nitric oxide can diffuse through the endothelial cell into the smooth muscle cells that are under the endothelium, vasculature, and they will activate the Guanine Cyclase, which will make the cyclic AMP, which will lead to relaxation. This right here, what you see right here, is not exactly a G protein. I don't think this is the G protein. The G protein is a different one, but it is not. The doctor didn't bring like any images for it in particular. Okay, but what you need to know is that the cyclic GMP pathway is associated with which G protein? It's associated with the 1213. Okay, that's the one that activates the GC, the guanylate cyclase. That's one. Two, it is associated with signaling that is associated that has nitric oxide. And three, what does it do? It activates protein kinase G, which leads to mainly smooth muscle relaxation and platelet inhibition. So it decreases platelet aggregation, which decreases basically thrombosis and clotting, basically. So this is why nitric oxide, which we basically take in as nitroglycerin, patients who have angina, they take nitroglycerin. This is turned into nitric oxide, which causes venodilation and relief of the pain, basically, that is associated with your angina. Aspirin is the only NSAID that actually results in the production of nitric oxide, which is why it is a staple in treatment of any coronary artery disease. Any coronary artery disease, you have to give the patient aspirin. There's no two ways about it. And almost anyone who has angina will be taken as well nitroglycerin. Okay? And as you can see here, it has vasodilation effects, anti-thrombotic effects, anti-inflammatory effects, anti-proliferative effects. So it's really good for keeping the health of your blood vessels. Okay. So with this, we are done with the G protein coupled receptors. And if you have any questions about these ones, feel free to ask right now. All right, we can move on to the next one, which is the RAS-G proteins. Okay, now RAS-G proteins are incredibly important. They are incredibly important for signaling pathways, cascades, transducers, all of these things. Now we need to talk about one thing, which is the Sh2 domain. What is an Sh2 domain? Real quick. Sh2 domain is a part of a protein, okay, that can bind to phosphorylated tyrosine residues. Okay, so when a protein has a tyrosine that is phosphorylated, in order to bind to this area, you need to have an Sh2 domain in your protein. What does a domain mean? It means it's just a part of the structure of the protein that has its own function. Okay, so the protein, as we know, it's like a lot of different parts together. Each one of them has its own function. And each one of these parts, we call it a domain. The Sh2 is one of these domains that its whole function is to be able to bind to phosphorylated tyrosine residues. Okay? Hope that's simple enough. Okay? So the RAS okay. So the RAS-G proteins, these RAS proteins, are not capable of binding to tyrosine to phosphorylated tyrosine. The RAS proteins, the RAS-G proteins, cannot bind to phosphorylated tyrosine residues. It requires an adapter to help it do that. And this adapter needs to have this Sh2 binding domain. Is this understandable? Is this clear? The Sh2 is in the adapter. So it is in, for the example here, it's in the GRB2. This is our adapter that allows the RAS to bind. Okay? Right. So these RAS proteins are incredibly important for growth factors. Most growth factors work through RAS-G proteins. Growth factors like what? Like PDGF, the platelet-derived growth factor, the epidermal growth factor, the TGF, all of these work through RAS proteins. So it's an incredibly important signal transduction, whatever, system. Okay. Again, they are regulated by GEFs and GAPs as well, because these RAS-G proteins work just like your other G proteins. When they have GTP, they are active. When they have GDP, they are inactive. So again, we have our GEFs and our GAPs. These are the ones that work. Okay. And again, they require an adapter protein, which is, as we said here, the GRB2. The adapter protein has to have what? It has to have an Sh2 domain. Okay. And now let's talk about this just image. Okay, so unlike the receptors for the G protein coupled receptors, here the receptor itself usually has its own activity. So the receptor here, whatever this receptor is, whatever it is, will have a tyrosine kinase activity. Now, what does a tyrosine kinase activity mean? It means that this receptor by itself will be able to phosphorylate the tyrosine residues on itself. So there's a tyrosine here in all of these blues. There is a tyrosine. So this receptor will phosphorylate itself. Okay? This arm will phosphorylate this arm, and this arm phosphorylates this arm. Makes sense? Cool. Now we have a receptor that has tyrosine residues that are phosphorylated. What do we do with these? Our RAS G protein wants to bind to it, but it cannot because it doesn't have an Sh2 domain. So an adapter protein will come in and bind to these phosphorylated tyrosine residues, allowing the RAS to bind through that. Okay. Now in comes this SOS. What is SOS? This SOS is our GF, the guanine exchange factor. We were talking about it before. Now this is one of the examples. There are multiple GEFs. This is one of them. It's called the SOS. All right. This SOS will bind with the RAS, and the RAS will bind to the GRB, and all of them will bound together to the tyrosine that is phosphorylated. When the GEF is bound to a RAS, what do you think will happen? The GDP will be kicked out, the GTP will be added. So now this RAS has a GTP. This RAS is another G protein, so a GTP will activate it. Now this RAS is activated. This activated RAS will go and cause phosphorylation. Will promote binding and phosphorylation of this thing called a RAF. All right, another name, a RAF. This RAF is a serine kinase. So not a tyrosine kinase. It phosphorylates serines. Okay, so that's why it's a different breed basically. Because it phosphorylates serine, it can now phosphorylate this MAPK, MAPKKKK, whatever. Okay, and it will go through the transduction, the signal transduction through that. So once the RAF, which is also known as a MAPKKK, is phosphorylated, it can go and phosphorylate the MAPKs basically. And this MAPK will go to the nucleus where it will activate different transcription factors, leading to transcription of different genes. Okay, so this is the RAS G protein, like the most basic pathway you can think of as a RAS G protein. Does this make sense? Is there any questions? Any problems? Okay, now the RAS G protein is very, very, very important in cancers. So many cancers will be caused by basically constant activation of the RAS protein. So basically what will happen is usually the GTP will, the GDP will never come back. So it will stay a GTP, and this will cause constant activation of all of this. Okay? And this is basically what happens in pancreatic cancer, most thyroid cancers, a lot of colon cancers. All of these can have RAS abnormalities, which is why over here I think the doctor mentions 30% of malignancies have problems in the RAS. The deactivator is definitely either the GTPase or the GEF. Both of these can work. So the RAS itself has a GTPase activity, and the GEF can exchange it. The GEF doesn't have to go one way. If you remember up there, where is the image? Yeah, right here, this image. It shows that the GEF itself can go both ways. Okay, it's generally speaking, it goes this way into GTP, making GTP, but it can go the other way if needed. GTPase is definitely one way. GTPase is always one way. From the name, it breaks down GTP. It does not form GTP. It only breaks GTP. So GTPase is going for GTP to GTP. Okay. Okay, so that's our RAS G proteins, and so mutations in RAS gene result in RAS proteins unable to hydrolyze GTP to terminate the signaling process, which will lead to malignancy. And that concludes our first topic, which is the G proteins. And I have the questions from the book. Now, I know nobody likes to read books, but Lippincott's book is a very simple book, and it is incredibly good and has everything, mostly everything. Like Dr. Ahmed has been trying to change the book that we use, Lippincott, into another book, because Lippincott is such a simple book, and he doesn't like how simple it is. He thinks that we should learn more stuff basically, and the university hasn't been accepting it. He's been trying for the past, I think, 2 years to change lipid cut. Kind of, yani, I honestly think that more people should use lipid cut because it's such a simple and easy book, and has and it has everything you need, and it's so visual, and everything is visual. The images are amazing, all of that. So yeah, again, I'm a big fan of the book, I guess. And it has good questions. So looking at this question, first question. AC is activated by a G protein. Which of the following secondary messengers will be generated? What's the answer? B. Okay. Next. All right. A manic depressive illness may result from the overproduction of IP3 and DAG and the accompanying signaling processes in certain CNS cells. Lithium is often useful in treating this illness. Lithium most likely functions to inhibit which of the following? So this disease is caused by overproduction of IP3 and DAG. Lithium will stop this disease. What do you think it does? It inhibits phospholipase C activity, of course, because phospholipase C activity increases IP3 and DAG. So if you stop that, if you stop phospholipase C, you decrease IP3 and DAG. Make sense? pKa would be CA. pKa is Adenine Cyclase. Adenine Cyclase, Cyclic Amp, protein kinase A. All right, we have here a 6-month-old male patient presents with slight fever, rhinitis, sneezing, cough. So he has pertussis. Okay, the toxin from this organism prevents the normal function of the inhibitory G alpha protein in cells of the respiratory tract. Which of the following disruption in cell signaling will result in the respiratory tract response to this infection? So which of the following will happen? So the inhibitory alpha is inactivated. Which of the following will happen? E, definitely E, overproduction of Cyclic Amp. We said both cholera and pertussis will overproduce Cyclic Amp, but this one will overproduce it by stopping the inhibitor. That one produces it by activating the activator further. Protein kinase A, which of the following is the correct answer? The cleavage of PIP2 is activated via GQ, induces the release of calcium. Activation by RAS-simulated gene transfer. This one I didn't mention exactly in the lecture, but you should be able to answer it by excluding by process of exclusion. It is D. Correct. It is D. It's not B. Induces the release of calcium from endoplasmic reticulum. What induces the release of calcium from endoplasmic reticulum? What was it? Do you remember? Which secondary messenger? IP3. IP3 will cause calcium release from the endoplasmic reticulum. Protein kinase A is a serine threonine kinase. It's not a tyrosine kinase. So D is the correct answer. Simulate the cleavage of PIP2. That's phospholipase C. Activated via GQ stimulation of phospholipase C. That's your the DAG, the protein kinase C. Activation by RAS stimulates gene transcription. It has nothing to do with protein kinase A. Last question from this lesson is, what is this? A constitutively overactive mutation form of RAS is present in cells from a breast biopsy sample. Therefore, RAS in these cells, so it is overactive. Which one of the following does the RAS actually do? E, it overstimulates the MAP kinase cascade, causing abnormal growth. That's what RAS does, right? So are we good with this lecture? Let me go to the questions for the next lecture. All right. Okay. There are just there's a few questions here. Woo. Okay. Let's prepare this and we go on to the next one. Do you guys want a break, a piss break, a water break, anything, or do you want us to just shoot through next? Chat. All right. All right. We'll keep going then. Ooh. Sorry, Caps. Let's go. Let's go. I don't need a break right now. Bismillah. So now we'll be talking about our next ones, which is mainly our tyrosine kinase receptors. Okay. Bismillah. So these are the catalytic receptors. The catalytic receptors are basically receptors that have like enzymatic activity inside of them. The receptor itself has an enzymatic activity. That's mostly what we mean by catalytic receptors. So again, here is just mentioning again the like self-oxidation. Yeah. So the receptor itself has an enzyme. Is an enzyme. Okay. So it can cause phosphorylation of stuff. That's what a catalytic receptor means. It has a catalytic domain. Here, just mentioning again the transmembrane signal. We mentioned this. The classifications again. We mentioned them. And now we will talk about this. Tyrosine kinase linked receptors. They are a receptor on the surface of the cell that is directly linked to an enzyme that is inside of the cell. This enzyme is a tyrosine kinase. What does it do? Phosphorylate tyrosine residues. Great, cool. Like what, for example? Growth factors. So what are the ligands, for example? Growth factors. The ligands like TGF, like epidermal growth factor, transformer growth factor, platelet-derived growth factors, all of these you will take in detail in the POD block next year. And you will basically take this lecture again and again, like 4 times in the coming year. For some reason, they always just go back to what a G protein is, what a tyrosine kinase is, the signal transductions. So you will have a lot of times to like remember what this lecture is. But yani, if you know it from the first time, you can skip it every single other time you get it. So the common structure here is our N-terminal is extracellular and binds to the ligand. Our C-terminal is intracellular and has a tyrosine kinase activity. And there is a transmembranous domain. And that is our typical tyrosine kinase linked receptor. Great, cool. The ligand will come and basically dimerize the whole receptor, and that's how it works. Receptors can be linked to stuff other than TKs, tyrosine kinases. It can be linked to it can be linked to serine kinases, threonine kinases, Guanine Cyclases, other phosphatases, stuff like that. Transmembrane domain means that it's a part of the protein that goes through the membrane. Transmembrane domain. That's it's just a way of saying that. Okay. Okay. So here we're just saying, okay, what are catalytic receptors? Again, tyrosine kinase linked receptors. That is the most common one. And then we have other ones which are like the tyrosine phosphatases, or the serine threonine kinases, or the guanylyl cyclases. Okay, all of these can be seen as catalytic receptors. Okay, now we will talk mostly today about tyrosine kinase linked receptors, and like mostly the most important one that is studied the most is the insulin one, and because diabetes. Okay, again here it's just showing you again how the structure is. N-terminal binds to ligand, C-terminal phosphorylates a tyrosine, and transmembranous domain. And this is just to show you how they look. There's a lot of different looks for each one of them. This is for the BDGF, VEGF, FGF, insulin, EGF, all of these. And as you can see, the insulin is a double dimer. It dimerizes and stuff. Okay, now let's talk about one other thing. So we are talking here about the catalytic receptors. So these are receptors that have an intrinsic tyrosine kinase activity inside of them. Okay, now we have other receptors that do not have a tyrosine kinase like activity that is intrinsic to them, but they are associated with other tyrosine kinases. So what do we mean here? We mean that it's just a receptor that once it dimerizes, so once a ligand comes and binds to it, as you can see here, so a ligand is bound to it. What happens is that this is not a picture of it. I think if I can find a better picture. Yeah, right here. So for example, this is one of the one example. I'm sorry for my bird. I don't know what is wrong with him right now. Anyways, so once a ligand hits the receptor here, the receptor will dimerize. Okay, and this shape is basically good for these other kinases to come and phosphorylate. Okay, so this is a receptor that does not have intrinsic tyrosine kinase activity. It requires an extrinsic and external tyrosine kinase for it to be phosphorylated, and then through its phosphorylation, different proteins that has an Sh2 domain can bind to it. Is this concept clear? The difference between a catalytic receptor and these receptors that do not have catalytic activities? Crystal, shut up. Okay. Right. So now we just to be clear, the TKs can dimerize. The TKs, you mean these ones? These ones, yeah, definitely. When a ligand will bind here, they will dimerize, and that's why they will start to work. They will only work because they have dimerized. And why do they dimerize? Because a ligand comes in. Okay. Okay. So now, what's the most important and the most commonly asked about tyrosine kinase that is external, non-receptor tyrosine kinase? The JAK. Janus kinase families, the JAKs. The JAKs are very important mostly because they are implicated in cancers of the blood, like cancers of the red blood cells and the white blood cells and the platelets. All of these cancers can be caused by JAKs. So that's why a lot of it's kind of important. Of these receptors, yeah. And here they're mentioning the SRC is actually is another tyrosine kinase that is outside of the receptor. The SRC and the JAKs mostly focus on the JAKs probably. Okay. This SRC is also found in cancers. And here it's just showing you again. So this is a normal catalytic tyrosine kinase. Ligand binds. Tyrosine kinase activity occurs. Tyrosine is phosphorylated. Proteins that have an Sh2 domain can now bind to it. Simple as we've been mentioning, tyrosine kinase, phosphorylated tyrosines, Sh2 bind them. Simple. Now, this is a slide that I have to say the doctor has kept in his lectures for the past 4 years since I was in first year. And since first year, I told him that the slide is incorrect. And he agreed that the slide is incorrect. However, for some reason, he still doesn't fix it. So what is incorrect about this? The slide starts off fine. The slide has, okay, so the adapter proteins contain highly conserved SH2 and SH3 domains. These are the domains that will bind into our phosphorylated tyrosines, okay? And then different receptors recruit distinct collections of SH2 containing adapter proteins. Now he wants to talk about our adapter molecules and then mentions RAS and STAT. RAS and STAT are not adapter molecules, or to be very exact, RAS is not an adapter molecule. We just mentioned what RAS is. What is RAS? RAS is a G protein that requires an adapter molecule to bind to phosphorylated tyrosines, correct? So it is not an adapter molecule. And as you can see in the book itself, if I go to the page where this was mentioned, you can find exactly, because the doctor basically copies from the book word for word. So I was really surprised when, you copy from the book word for word and yet you make a mistake. How does that work out? And it's right here. So this is the sentence that he copied. Adapter molecules can function in single energy by different growth factors. Certain adapter molecules are known to be critically important in multiple signaling pathways. And this is the point that is mentioning of important adapter molecules, and it mentions the RAS as in the RAS pathway has these adapter proteins which contain the SH2, which we mentioned were the GRB2, if you remember. There, the adapter molecule was the GRB2, which contained the SH2. So here they're just mentioning, oh, in this pathway, the adapter molecule is X. And then in the stat, it mentions, and yes, the stats, they are adapter molecules. That is correct. The stats are adapter molecules because they contain the SH2, but the RAS is not an adapter molecule. Okay? Is this understood? Because it is a mistake that the doctor, he told me, and yes, it is a mistake. 4 years ago he told me, and 4 years later he did not. He just has to remove these. Yeah, the pathway. So for the RAS, it's just mentioning in this pathway you can find adapter proteins, and the RAS itself is not an adapter molecule. Okay. Ooh. So all right. While the stat is definitely, I think the stat is an adapter protein just because it has an Sh2 domain, as we will see further. Okay. Okay. So again, what is an adapter molecule? Adapter molecule is something that contains Sh2. The Sh2 is the SRC basically. That's our adapter molecule. The RAS, we said it has the adapter molecules, which where is it? The RAS right here, it was the GRP2. That's our adapter molecule here. In the JAK-STAT pathway, it is the STAT itself. That's our adapter molecule. Cool, great. Now, we already talked about the RAS G protein pathway. Not going to repeat it, but I will be talking about what the JAK-STAT pathway is. Again, this is a pathway that uses JAK. So do you think it has a catalytic receptor or a non-catalytic receptor? What do you think? Non-catalytic. Why does it have non-catalytic? Because the JAK itself is the enzyme here. The JAK is the kinase. We don't need another kinase. Actually, we need the JAK because we don't have a kinase. Okay. So here, what happens? What happens is ligand binds, receptor dimerizes. Now it is ready to be phosphorylated by JAK. JAK comes, and JAK is a tyrosine kinase, so it phosphorylates the tyrosine residues on this receptor, this non-catalytic receptor. Once they are phosphorylated, the SH2 containing stats. What is a stat? A stat is a signal transducer and activator of transcription. So it's a signal transducer. It amplifies the signal, but also it is a transcription factor. It is both. It's a transcription factor that contains an SH2 domain. Okay, we know what is transcription factor, right? Transcription factor is a protein or something that will induce transcription, induce mRNA formation from a certain part of the DNA. So the stat is a transcription factor that contains SH2. Okay, this stat, which contains SH2, will bind to the phosphorylated tyrosine residues on this arm. And once it is bound to it, the JAK will come and phosphorylate it as well. Once the stat is phosphorylated, it will dimerize with the other stat that is on the other arm that has been phosphorylated by the JAK on the other side. There should be a JAK here as well. And they will dimerize together, and a dimerized stat, double stat, will go together as a transcription factor into the nucleus, transducing causing transcription of different genes. And that is our JAK-STAT pathway. Is this understood? Yes. So the JAK is a tyrosine kinase, and the stat requires a phosphorylation on its tyrosine residues for it to dimerize with the other stats and translocate to the nucleus and stimulate transcription of target genes, blah, yada, yada, yada. All right, that's our JAK-STAT pathway. The JAK-STAT pathway is with non-catalytic receptors that require a phosphorylation of their tyrosine by a JAK. All right, and the JAK is a kinase. Cool. Yeah, so with this, we are off to the next pathway, which is the Pi3 kinase pathway, the Pi3K pathway, not to be mistaken for the PLC pathway. Okay, I don't know why I always had a problem with which one is the Pi3, which one is the PLC. The PLC is with G proteins, the Pi3 is with tyrosine kinases. From the name, it has a kinase. It's a Pi3 kinase pathway. Crystal, I would kill you. Okay. Oh my god, this bird. Okay, so this one works very similarly to the other ones, but this one actually needs a catalytic. Over here, you have a catalytic tyrosine kinase receptor. Okay, so ligand binds, again, simple, ligand binds, tyrosine phosphorylated. Now we have this Pi3. This Pi3 kinase will bind to these phosphorylated tyrosine residues. Okay, it will bind to them, and this is what activates them. Now, do tell me, this Pi3, exactly, great, this is exactly what I was going to say. This Pi3 kinase binds to tyrosine, the phosphorylated tyrosine. So what do you think this Pi3 kinase has? What domain does it has? It has a Sh2 domain. It has to have an Sh2 domain. So the Pi3 kinase has an Sh2 domain, which allows it to bind to the phosphorylated tyrosine. Cool? Now it's bound to this. This binding somehow activates it for some reason, and when it's activated, it goes to the membrane and goes to the PIP2s. Now, in the phospholipase C pathway, we cleave the PIP2 into an EIP3 and a DAG, right? Here, the PI3 kinase, it is a kinase. What does a kinase does? What does a kinase do? It will phosphorylate the PIP2 into a PIP3, so adds a phosphate to it. Now that we have this PIP3, it is capable of activating the AKT, which is a protein kinase B, and this will lead to the activation of BAD. BAD is one of the apoptosis inhibitors. I think you guys took cell death, and in cell death we have like the BAX and the BAC and the BCL and the BCL, these ones. BAD is one of them. It is a apoptosis inhibitor. So it promotes cell survival. That's what BAD does. Okay. So that is the main pathway. The kinase is activated by the phosphorylated tyrosine. It phosphorylates PIP2 into PIP3. PIP3 activates AKT, which is protein kinase B. This protein kinase B will activate BAD. BAD will promote cell survival. Now, how do we shut down this pathway? Through a molecule called PTN, phosphatase and tension homologue. It is a phosphatase. It goes to PIP3. What do you think it does? It will remove the PIP3, making it a PIP2, which shuts down this pathway. Okay, so PTEN is an inhibitor of the Pi3K pathway, right? The PTEN is indicated, or like problems in the PTEN. If a mutation happens that inactivates the PTEN, cancers happen. Specifically, endometrial cancers are mostly caused by PTEN mutations. Okay, and activations of the PTEN causes uterus cancers, endometrial cancers. Okay, is the Pi3K pathway clear? So in the Pi3K pathway, if someone asks you, what do you think is the secondary messenger? What would you say? Which one of these is a secondary messenger? No, not the protein kinase B. Remember what the secondary messenger was in the other pathways, and apply it here. The other pathways, cyclic AMP activated the protein kinase A, and the cyclic AMP was secondary messenger. So here, the one that activates the effector was the PIP3. So yes, both of you are correct. The PIP3 here would be the secondary messenger because it is the one that activated the effector enzyme, which was the protein kinase B here, protein kinase A there, protein kinase C there. The ones that activates these are the secondary messengers. Okay. And the secondary messengers are basically the products of the initial enzymatic reaction. So the initial enzymatic reaction was Pi3K turning PIP2 into PIP3. Again. Okay. So that's our Pi3 kinase pathway. Now our last tyrosine kinase pathway we will be talking about is our lovely insulin. Yay. Insulin. Insulin signaling occurs again through tyrosine kinase activity, and this one has intrinsic tyrosine kinases. It is a dimer, so it has a 2 beta, 2 alpha. These two, once the insulin comes here, they will dimerize together, and this will lead to tyrosine kinase activity, and the usual, basically the usual. Okay, the insulin receptor itself, our joint primary, it contains a Sh2 domain. Yeah, and basically it will phosphorylate itself. The tyrosines will be phosphorylated, and then this IRS, which is the insulin receptor substrate, will be phosphorylated by the kinase. And once the tyrosine residues on the IRS are phosphorylated, this IRS will go and activate all the signaling pathways that are associated with insulin, like basically, yeah. So it will cause glucose uptake, glycogen synthesis, protein synthesis, fat synthesis. Everything you took in MOL for, until now basically works through this signal transduction. Yeah. Yeah. So, and he's telling you here, the IRS alone can activate RAS, can activate STAT, can activate Pi3 kinase. So it will lead to so many different, basically, results. Okay. One of the main things that it does, as you can see here, is the GLUT4. If you remember, GLUT4 is the glucose transmembrane, or like the, what is it called? Channel, the glucose channel that is found on, I think, RBCs and no, not RBCs, muscle cells. I don't know, muscle cells. It's on something else. I'm not sure. Muscle cells and something else. I think fat cells. Was it fat cells and muscle cells? Either ways, it is the one that is present on muscle cells. So if you do not have GLUT4, the muscles cannot use glucose, basically. And yeah, okay, good. So I was correct. Yay. And which means, and this GLUT4 is why is it expressed? It's expressed as a result of insulin binding to the receptor. So the insulin binding to the receptor will result in the GLUT4 being expressed on the surface. So without this transduction, GLUT4 will not be expressed. Cool, great. This is just an arithmetic that the doctor also made for this whole thing. Give us a second. So here we have our receptor, which has been, the tyrosines has been phosphorylated. They now phosphorylate the IRS on the tyrosines. So it has been phosphorylated here, and now it is activated, and it can activate the Pi3K pathway, which activates the AKT, which will lead to pro-life stuff, which activates basically anti-apoptotic effects, and among them is basically giving your cell more energy. How do you give cell more energy? Letting glucose go in. So this is the idea of this. This only happens if the tyrosine residues of both the receptor and the IRS are phosphorylated. Now, what happens in insulin resistance? Give me a second. Someone is banging on the door. Guys, I'll just have to go for like 3 minutes. I need to move my car. I parked in a wrong place. Lol. All right. Yeah, recording back. Okay. Now, glucose causing inflammation regardless of this. Now, the point that the doctor was saying here. Okay. Now, this is what happens basically when you have the insulin resistance happening. Okay. So insulin resistance Happens because of many things. But mostly what we're looking at is inflammation. Okay, insulin resistance as a process is mostly caused by residues other than tyrosine being phosphorylated. So not phosphorylating tyrosine, specifically serine and threonine phosphorylation. So when the serine or the threonine of the receptor or the IRS are phosphorylated, that's when we have insulin resistance, because insulin cannot continue with its pathway if the tyrosine is not um, phosphorylated. Okay, so I don't think the doctor really meant glucose causes inflammation. It's more of eating. Eating in general, anything foody. Okay, eating in general is noted to increase inflammation in the body, especially high caloric intake. High caloric intake increases um, inflammation in the body. Specifically, vegetable oils will very much increase inflammation in the body. Okay, inflammatory markers, inflammation. Okay, so what happens? It's not a mistake. Okay, it is not really a mistake. It is the result of these, the TNF alpha, the tumor necrosis factor alpha, and the IL-6. These receptors are their normal pathways involve serine threonine phosphorylation. So their normal metabolic pathways will involve serine threonine phosphorylation. The thing is, in the body, in the cell, there's no, okay, I will only phosphorylate this protein, I will not phosphorylate that protein. That is difficult for the cell or for some enzymes to decide. Whatever they can phosphorylate, they will phosphorylate. So when you have an increase in inflammation in the body, these ones, the TNF alpha and the IL-6, will be elevated, and these ones will of course lead to serine and threonine phosphorylation via this SOX. Okay, this is their secondary messenger, or this is their effector protein, the SOX3. And the SOX3, then its normal activity is to phosphorylate serine. It's the normal activity of this protein. It is something that your body requires, is serine phosphorylation. But when it is overdone, what happens is that it will start when it's like constant all the time, you have inflammation in your body, you will find that your insulin receptors are now also being just entirely blocked because oops, they are phosphorylated in the wrong place. And this happens mainly in cases of obesity, in cases of increased adipocytes, or like systemic inflammation in general. Like systemic inflammatory diseases can also lead to diabetes, because again, you always have inflammation happening, and this can lead to insulin resistance. Okay, but who the thing is, not food and eating a lot of food, high caloric intake itself can lead to insulin resistance by inducing inflammation. Yeah, and for insulin, the serine should not be phosphorylated. The serine is just one of the 20 amino acids, right? So of course it has the insulin receptor will have all the 20 amino acids. It's normal, yani. And the serine is not supposed to be phosphorylated in the insulin receptor nor in the IRS. The tyrosines are the ones that are supposed to be phosphorylated. But what happens is, because this IL-6 and TNF alpha are are prone to serine phosphorylation, and they cause serine phosphorylation, they will phosphorylate what shouldn't be phosphorylated. Okay, and حتى it's not that it should not be phosphorylated. Like at times you can imagine that when you have inflammation, okay, in your body, just think about it this way: if you have inflammation in your body, do you think you want to keep your glucose, or do you want to keep your energy basically, or do you want to use it? Most likely you will be wanting to actually keep the glucose. You don't want to use your glucose because you're having a disease, so you want to save your glucose. For example, you want to not let your muscles use it. You want to use you don't want your lipids to use it. You want to use your glucose in other places, like your neutrophils, your macrophages need their glucose, need your energy. So it is, you need to understand that this is not by mistake. The fact that the TNF alpha and the IL-6, they will stop the insulin from working. This is not a mistake. This is what it is supposed to do, because I don't want the insulin to be working in the state of inflammation. I want the insulin receptors to work I want the glucose to go to other places other than my muscles. I don't need my muscles. I don't need to, go on an exercising when I am sick and inflamed, So this is basically a protective factor in our body that stops our glucose from being used in wrong ways while we are sick. But the problem is that this inflammation also occurs just by plain eating or by being obese and stuff like that, which is why insulin resistance can occur in states other than being in a disease or such. Okay, so always try to think, why is this the case? Why is TNF alpha and IL-6, why do they stop insulin from working? Because the body isn't really stupid. It knows what it's doing, but at times it can overcorrect, basically. And this is what happens here. It's overcorrecting, which is leading to your insulin just not working anymore. Okay? Does this make sense? Yeah, free fatty acids. Okay, so this is a very simple way of knowing, of understanding why free fatty acids themselves can be turned into inflammatory mediators. Like fatty acids, they can turn into inflammatory mediators, like prostaglandins. So by turning into prostaglandins, they lead to inflammation, and inflammation leads to insulin resistance. And this is one of the ways. There's more than one way, but saying this is just one of the ways that I can remember right now. Okay, and that concludes our second topic, which was the catalytic receptors, tyrosine kinases. I will open the questions, and you guys can think there should be easy, inshallah, for you guys. Whoop. All right. We have here our first question. Prolactin stimulation of its target cell begins by its catalytic receptor catalyzing production of secondary messenger, dephosphorylating serine threonine, forming dimers in the membrane, stimulating tyrosine phosphorylation of RAS. I'm not sure. Do you guys know this? Okay. So again, it's a catalytic receptor. So which of the following is probably the correct answer? If it's a catalytic receptor, think of it like that. Yeah, it should be D. It forms dimers, and then from that, so it's a catalytic receptor. Catalytic receptors will work. First of all, they form dimer, and then they start working, right? They don't activate G proteins. They don't catalyze production of secondary messengers. They don't have CAMP and all of these. That's not like their first step. Dephosphorylating serine threonine residues, we didn't come across any enzyme that works through that. And stimulating tyrosine phosphorylation of RAS, that is untrue. RAS does not get aslan phosphorylated. RAS is a G protein, doesn't need phosphorylation, right? Okay, let's see this question. Interleukin 2 binds its catalytic receptor on a T lymphocyte. So we have a catalytic receptor. In response, which of the following will change within the cell? What do you think? I think this should be, again, I'm not sure about this one either, but I think it should be C, right? It is C. Yeah. The levels of phosphotyrosine within a cell will change in response to catalytic receptor signaling. Secondary messengers, including calcium, will not be altered by catalytic receptor signaling. Yeah, we already mentioned that. And Adenine Cyclase is an enzyme linked to some G protein. Its activity will not be activated by catalytic receptor signaling. So yeah. Other question. Stats function in signal transduction by activating GTP binding to the alpha subunits, binding receptors phosphorylated on serine threonine residues, linking to G protein coupled to transmembrane, phosphorylating substrates on tyrosine residues, stimulating transcription of responsive genes. What do you think this one is? I think it's B. E. Yeah, STAT is a transcription factor, so it should be E. It stimulates transcription of responsive genes. Insulin binds to an insulin receptor. What's with B? B, binding receptors phosphorylated on serine threonine residues. What was that? Did we mention anything that has serine threonine residues that we want to? I think it was RAF. Yeah, RAF is a serine protein kinase that will phosphorylate MAPK on serine, right? So RAF would phosphorylate the MAPK on serine. All right. Next question was insulin binds to an insulin receptor of an adipocyte. Which of the following is a signaling process that will occur in response? What do you think? This one should be easy. Yes, it is E. The tyrosine phosphorylation of IRS. E. I think this is the last question. Yeah. A cell has a mutant form of PTEN. PTEN. As a result, which of the following signaling molecules will remain active longer than usual? Which one was it? What do you think? It was C. Yeah, PTEN is associated with the Pi3 kinase pathway. And that's our second lecture. Do you guys need a break or do we go for the last one? The last one should be the shortest, if I remember correctly. The doctor removed the biggest part of the lecture from our year. He had this whole thing. Did he teach you this thing? The whole cholesterol turning into progesterone, cortisol, testosterone, and the congenital adrenal hyperplasia, all of these things. Nope. Okay. Wow. He skipped the entire congenital adrenal hyperplasia. Okay. Wow. Actually, wow. Anyways, I'll just zoom in here. Okay. So you guys want me to keep going, or do you want to take a breather? Yeah, I'll finish before 7, inshallah. 7 max, bi'idhnillah. Okay. All right. Our last topic will be steroid signal transduction, steroid signaling. Now, steroid signaling is different from the signaling we've been mentioning until now. Until now, the signaling we've been mentioning is instantaneous, or like it's by hours max, minutes, usually seconds to minutes. So what happens is ligand binds, everything happens, and everything happens basically. Protein kinase activates phosphorylation of proteins. Proteins that are phosphorylated will either work or stop working. These stuff. And the only exception to that is the JAK-STAT pathway, because the JAK-STAT pathway works as transcription factors. And in general, transcription and the translation of proteins, takes a while. So why the difference? It's just because the end product of the ones that we've been talking about, it differs. So the end product before was just, a protein has been phosphorylated, now it's working, done. That takes like minutes. Here, the end product is the formation of a whole new protein after transcribing a whole new mRNA from a DNA. So that's why it takes longer for steroid signaling and for stuff that need transcription factors at the end. It will take longer for them to work. Okay. So again, he here again talks about the steroid hormone, peptide hormones, amino acid derivative, prostaglandins. I think we all know what these are. Now we can talk about our steroids. Steroids come in 3 main shapes, since 3 main kinds. 4, you can say 4 honestly. But we're talking about carbohydrate regulations, which are the glucocorticoids, mineral balance, which is the mineral corticoids, and the reproductive functions, which is the gonadal steroids, the testosterone and progesterones and the estrogens. Okay, all of these are formed in your adrenal glands and the sexual glands, so the testes or the ovaries. The testes and the ovaries only form the reproductive stuff. The adrenal glands will form everything. So they give you the mineralocorticoids, the glucocorticoids, and the sex hormones. Okay, all of these are you can get in your adrenal glands. Cool, great. Steroids play a role in inflammation, stress responses, bone metabolism. Yeah, so not only do we have these three, the glucocorticoids, mineralocorticoids, and gonadal steroids, but also your vitamins, so vitamin D, vitamin A, vitamin E, vitamin K, all of these are considered in your kind of steroid hormones. They're not exactly steroid, but they are associated with your steroid hormones because they work the same way, because they are all hydrophobic. Okay? So our steroids, again, glucocorticoids, mineralocorticoids, androgens, which are the testosterone, estrogens, the estradiol, estron, estriol, and the progestins, which is mainly progesterone. Right? These are our steroid hormones. All of these are derivatives of what? What is the derivative? What makes all of these? It is cholesterol. Cholesterol is extremely important to make all of these. Okay? Now here he just mentions a few shapes of nuclear receptors. Now why are we talking about nuclear receptors? Because all of these work on nuclear receptors. Okay, what do we mean by that? We mean that what was there? Oh, the picture was way far back. But as you can see here, these work by going all the way to your nucleus and working on the DNA right there. Okay, so this is how they work, and we'll be talking about that more right now. The one we want to focus on the most is this one because it has all the steroids. You can see glucocorticoids, mineralocorticoids, progesterone, androgens, estrogens. Everything you basically care about right now is here. Here you can see vitamin D is here. The thyroid hormone is here. The prostaglandins here. And a few other things that we don't know what they are, as you can see here. Lovely. Okay. And retinoid. Where is retinoid? I'm not sure where retinoid is, but retinoid is what? It's vitamin A. Okay. I think it's here. Yeah, that's it. Retinoid. I think that's it. It's here. Anyways, so let's talk about our intracellular receptors. Okay, so steroids, their receptors, where are they found? We said everything we've been mentioning until now has been membrane-bound receptors. So the receptor is on the membrane. Not a steroid, but functions the same. Retinoid, yeah. Vitamin A, vitamin E, vitamin K, and vitamin D. All of these are not steroids, but they function like steroids because they are all fat soluble. They are all hydrophobic. Retinoids are just vitamin A. Vitamin D is cholecalciferol, and these ones. Okay, so intracellular receptors. Until now, all we've been talking about has been membrane receptors. So they all had like their N terminal was outside, their C terminal was inside. There was a transmembrane domain. All of them have been the same, right? Now we have this, which is different because they are already inside the cell. These ones are intracellular receptors. They are inside the cell and they are waiting for the steroids to enter the cells and come to them. Okay? All of them are similar in structure to a point where they have four main domains. Okay? Again, we were mentioning what are domains. They are just parts of the protein that have a specific function. So the domains we're talking about, first is a variable domain. So what do we mean by variable domain? It is the domain that works and interacts with the transcription factors. So this is the effector area. This is where the effect will come in. And this is near the N-terminal. Then we have the DNA binding domain. This domain will find the DNA it wants to go to. It will be able to read the DNA and bind to where it wants to bind. How does it do that? Through motifs called the zinc finger motifs. These zinc fingers can basically read the DNA, read the nucleotides, and know, okay, this is where I want to stop, this is where I want to bind. The third domain is domain for dimerization. If the receptors need to dimerize, this is the area they dimerize at. And lastly, the fourth domain is the domain where the steroid will bind to. So unlike in our membranous receptors, There, where was the ligand binding? The N-terminal or the C-terminal? There it was the N-terminal. Now, our hormones here are binding to the C-terminal. Just make sure you keep that in mind. 1 and 4 are completely different because 4 is where our hormone, the steroid, is binding. 1 is what is the part of the receptor that's actually acting on the transcription factors. This is where the receptor basically plays with the transcription factors and activates transcription factors or stops transcription factors from working. Okay? Now, this is again the same structure we were mentioning here, but now it's more like folded up. N-terminal, this is where the transcription factor interaction happens. The DNA binding domain, which has the zinc finger motifs. The C-terminal, which is where the hormone binds. And here is the middle region where it can dimerize, it can hinge, it can do other stuff, like anything that is needed for its structure to work. This is where it happens usually. Okay. Now, one thing that is mentioned here, again, the zinc finger motifs, we mentioned it before. What are they? They recognize and read the DNA nucleotides. That's why they are important. The other thing I want to mention is this, the nuclear localization signal. I think you guys took cell trafficking or protein trafficking last semester, correct? In protein trafficking, we mentioned different signals that can localize to different area in the cell. So some proteins can go to the mitochondria, some proteins can go from the endoplasmic reticulum outside into the endoplasmic reticulum, and some proteins can go into the nucleus. This is what we're mentioning here. All the steroid receptors need to have the signal that allows them to go into the nucleus. So all of them have the nuclear localization signal. If you remember that concept from protein trafficking, all of them must have it to be able to go into the nucleus, because all of them want to work where? On the DNA in the nucleus. Okay. Now here he just mentions a bit about our the zinc finger motifs. The motif basically, so he starts wants to explain basically what's the difference between a domain and a motif. And the main thing you need to know is that a motif is a structure, while a structure caused by basically different secondary structures, 2 or more secondary structures. We took the secondary structures like beta sheets, alpha helices. These ones are the secondary structures, right? So more than one secondary structure can form a motif. Okay, that's why we call them super secondary structures. So motifs are names for a structure and not for a function. Motifs do not have to have a function. They're just looks. They're just how they look. They're just a structure. On the other hand, a domain is an independent unit that has been folded in a certain way so that it can have a specific function. So a domain is associated with a function. A motif is associated with just a structure. How does it look like? What does it do? Okay? But of course, domains are generally made of different motifs. Yes, they are the ones that recognize the DNA, true. But when we say zinc motifs, that way, just us saying, oh, they are zinc motifs, we're not talking about their structure, their function. We're just saying, oh, they are a structure that has zinc in them, zinc finger motifs. And then, as you can see here, it is called a DNA binding domain. So the domain describes a function. A motif describes a structure. Is this? I think this is the best way you can understand it. Of course, there is difference between them, because one of them is just multiple structures. This one is folding that results in a certain function to be able to be done. But the main thing you need to know is domain is used when we're describing functions. Each domain has its own specific function. Motifs will have it's just a structure. Will it have a function? It doesn't have a function. It is irrelevant. What's relevant is that it's a structure. Okay. So our DNA binding domain must have a zinc finger motif, because the zinc finger motifs allows the DNA binding domain to do its function. Okay. Most proteins involved in cell signaling consist exclusively of small domains expressed by linker joints. As you can see here, we have 4 domains in this one receptor. One domain described by its function, playing with other transcription factors, by the function binding domain, and by the function dimerization, by the function binding to the hormone. Okay? Each one of them are domains. So that's it for our motifs and our domains. And now we talk about the classification of steroid receptors. Steroid receptors come in 2 main types. One of them is free swimming in the cytoplasm. These are type 1. And type 2 is bound to the DNA at all times. This is type 2. So type 1, the receptor itself is freely swimming in the cytoplasm. Okay? It is usually associated with the heat shock proteins. So they can, heat shock proteins, if you remember, they allow different foldings basically. So they are associated with heat shock proteins to allow them to go into the nucleus. Okay, and change their folding, allow them to go into the nucleus because they are in the cytoplasm originally. Okay, while the type 2 ones are already in the nucleus, they are already bound to the DNA, and they are just there waiting for the steroid to come and activate them. Okay, now an interesting thing is that the estrogen receptors are nuclear receptors, right? So they are present in the nucleus. However, they are considered type 1. Why are they considered type 1? Because they are not bound to the DNA. Okay, so they are present in the nucleus, but because they are not bound to the DNA, we consider them type 1 receptors and not type 2 receptors. Okay, that's just one thing you should know about so they are swimming in the nucleus, the nucleoplasm, not bound to the DNA. They're just swimming around in the nucleoplasm and not the cytoplasm. Okay. Okay. So again, here they're just mentioning the examples for the type 1, our sex hormones, our glucocorticoids, and our melanocorticoids. So everything that comes out of your ovary, testes, or your adrenal glands, all of them are type 1 receptors. The rest are type 2. So your thyroid, your retinol, your vitamin A, your vitamin D, your vitamin E, K, all of these are type 2. The active receptor complex binds to DNA through the... Okay, so this is just good descriptions for them. Okay, so the receptor complex, okay, the receptor plus the hormone will go from the cytoplasm in the type 1. They will go from the cytoplasm into the nucleus, where they will interact with hormone receptor. I forgot the word. Response element, with hormone response elements. Okay, those hormone response elements are present on the DNA, and basically it will cause the effect of the transcription factor, the changes in the transcription factor on the DNA. Okay, so what they're saying here basically is that type 1 receptors are present in the cytoplasm or the nucleus. The important thing is that it's freely swimming. The steroid will bind to them, and then they will go and find their response element, the HRE, their response element that is already present on the DNA. They go there, they bind there, they do their work. Okay, so they bind on their response element. The other form is the type 2 receptors. The type 2 receptors, okay, you always have the receptors and they are always there on the DNA. They're always bound to the DNA and waiting for the steroids. Okay, and why are they waiting for the steroids? Because they are always bound to a repressor. They are always bound to something that is repressing them, that is inhibiting them. Once a steroid comes and binds to them, this co-repressor will stop working, so these receptors will start to work as they should, and they will start causing transcription and such. Okay? Are hormone response inherent parts of type 2, or are they only needed for type 1? They're only needed for type 1, hormone response elements. Why? Because hormone response elements are basically the the place where like the plane lands. I'm not sure what the word was in English. بس يعني المهبط الطيارة, So imagine that the complex is the plane, and the hormone response element is basically saying land here. You want to land here. This is where you want to work. So of course the hormone response the hormone complex will go, the receptor complex will go and then bind to the hormone response element over there. On the other hand, with type 2, you don't need anyone to tell you where to land because they're already bound to the DNA. They don't need to be told, oh, you need to land over there, because they're already bound to the DNA, right? But the receptors are always bound to these repressors, and these repressors will be inhibited when a steroid binds to them. So for these, we're talking about vitamins. For these, we're talking about the steroids. Okay? Is this clear? Lovely. Again, this is just retelling of what we've been saying until now, but it has one small extra thing that you need to keep in mind. Okay, so steroids, we said they are hydrophobic, so they enter through here, no problem. Blah, blah, blah. They go cytoplasmic receptor, and then into the DNA, they work, and they start the transcription, translation protein. This is what we've been talking about until now. But now there is one other thing we kind of want to know, which is that the steroids also have membranous or membrane receptors. Okay, they are much less, and they are much less important, or like they have less important effects, but they cause rapid responses because they work through the effects that we've been talking about until now. Like they work through G protein, they work through tyrosine kinases, they work through JAK-STAT, all of these things. Okay, so once the same steroid, it can go into either the cytoplasmic receptor or the membrane receptor for it, and this will lead into different results. These ones, it will lead to the stuff that we've already been talking about, to a rapid response that takes seconds to minutes, while this one will take hours to days. Okay, and both of them are important for steroid functions. Okay, and this is the difference between. So one of them is called a nuclear initiated steroid signaling, which is this one, the number 2. Are they just are used to insert, or any time serum wants to find these faster responses are always there, and the steroid needs them for their function. They need them for their functions normally, but these rapid responses do not include transcription or translation or making new proteins. No, they're just like what we've been talking about until now. They're just, I don't know, increasing calcium, phosphorylating proteins, yada yada. These rapid responses, like here, stimulation of protein kinases, activation of G protein, change the intracellular calcium. While the nuclear initiated steroid signaling are the ones that are associated with protein synthesis and gene transcription, classical signaling, rapid signaling. Okay, both are important for normal function of steroid hormones. Both are important. All right. Again, he is mentioning the same thing over here. Latency of seconds when we're talking about membrane. Latency of hours when we're talking about intracellular. The cellular responses, this is in vivo response, usually transcription, translations, whatever. Okay, so what is this saying? Estrogen receptors, type 2 receptors are always in the nucleus. The estrogen and progesterone receptors are in the nucleus, however, they're freely swimming. These ones are always connected to... No. The rapid signaling is not going to... It is going to make its own products, but not the product which is formed by the NES. Okay, the MES, which is the membrane initiated ones, will make its own products, which is G proteins, cyclic AMP, protein kinases, whatever you want to say, the things that we've been talking about for the past 2 lectures. This one will cause it normally, so its products will be created fast, rapidly, but its products are not proteins, are not protein synthesis and gene transcriptions. This is the responsibility of the NES. MAPKKK does transcription too as well. Yeah, the MAPK pathway will lead to transcriptions, and these transcriptions will take a while to happen. but the kinases themselves will cause, phosphorylation of other proteins as well. Okay. Yeah. So the MIS, rapid response, and everything from G proteins, calcium, protein kinases. The NIS is the whole gene transcription, protein synthesis, and the MIS is on the membrane. It causes the usual stuff that we've been talking about. The steroid one is in the cytoplasm or the DNA or the nucleus, and it causes transcription of different DNAs. Okay, so here's telling you, in the absence of hormone, our estrogen and progesterone receptors, where are they? In the nucleus. Are they bound to DNA or freely swimming? Freely swimming. Type 2 receptors are in the nucleus. Are they bound to DNA or freely swimming? Bound to DNA. The glucocorticoid and androgen receptors, where are they? In the cytoplasm, and they are freely swimming. That's in the absence of hormone. In the presence of hormones, all of them will just go to the nucleus and bind to the DNA and make the magic happen. That's it. In the presence of hormone, they will stimulate the receptor, they will translocate into the nucleus, they will bind to their hormone response elements when we're talking about estrogens and glucocorticoids and progesterones and androgens, and they will activate gene transcription. And in the case of type 2 receptors, once the steroid binds to them, it will inactivate the co-repressor, which will lead them to start working. This is here, it's mentioning how the MIS actually works. They work in these caveoli, which is basically like little caves in the plasma membrane that has the receptors which will bind to the hormones, and this will lead to activation of kinases, PKA, PKC, PLC, MAP kinase, RAS, all of these, which will lead to more rapid responses, like ion channels, protein kinases, phosphatases, but also it will lead to translation transcription, which are not as rapid, and this is the work of RAS and MAP kinase. Okay. Yeah, they are the MIS effects are faster than NIS. Why? Because they do not require synthesis of proteins. Examples are epidermal growth factor receptor. Its activation can result in sustained MAP kinase signaling. Okay, and of course there will be crosstalk between the NIS and the MIS. It's not just NIS nor just MIS. The NIS might affect the MIS somehow. The MIS can affect the NIS somehow. All of them can happen. Last thing I think he mentioned here is just that the hormone response elements that we've been talking about are specific for hormones. So the cortisol or the glucocorticoids have their own response element. The mineralocorticoids have their own response element. The androgens, the estrogens, the progesterones, the vitamins, yeah, all of these will have their own hormone response elements that they go to and they activate. Okay, and these hormone response elements can be before the gene they regulate or after it. It doesn't matter. Is it upstream? Is it downstream? What matters is that it is near to the transcription complex, or it's near to the activation of the transcription factor, and they lead to the activation of it. To MAP kinase, then it does include, yeah, of course. MAP kinase leads to transcription factor stuff. So yeah, it does include protein synthesis, which is why it does take more time. So this transcription translation part right here should not be rapid. GRE, maybe look it up. Okay. Response times for ion channel receptors are measured in milliseconds. Action potentials require like milliseconds of fastness. For GPCRs, transfer enzymes, JAK-STAT receptors, all of these in seconds to minutes. طبعا we're talking about here JAK-STAT, the time it takes for the stat to go from the cytoplasm to the nucleus, and then the stat will start, causing transcription, which will take a long time. But here we're talking about the time from the activation of the receptor to the stat starting transcription is seconds to minutes. And then for steroid receptors, it's hours to days. And that is it for hours to move. No, hours to start the for the transcription to finish. For the transcription and translation to finish. Hours to days. And that is the lecture. Now just the last few questions. A particular cell type under study is known to be the target of a certain hormone's action. The hormone has an intracellular receptor that normally resides within the cell's nucleus. The most likely identity of the hormone is B. Estrogen. Estrogen is found in the nucleus, however, it is a type 1. Remember that it is a type 1. Right. Cells isolated from a breast tumor and grown in cell culture in the laboratory are found to respond to estrogen via both intracellular and membrane-bound receptors. Which of the following is most likely to result from signaling via the intracellular receptor? Synthesis of estrogen responsive proteins. Yeah, synthesis of proteins. That's what matters. Yep. All right. A 43-year-old female patient has an estrogen receptor positive breast tumor. And SERM means a steroid. Basically, it's a estrogen receptor antagonist. Basically, okay. So an estrogen receptor antagonist is used as treatment. The beneficial actions of this drug to the patient result from the drug. No, it's you don't want this. Wait. This is an estrogen receptor antagonist. So it stops the estrogen receptor because this, yes, it is C. It will be C here, blocking the binding of estrogen to estrogen receptors. Okay. Because these cancers basically have so much estrogen receptor that it is causing too much protein, protein synthesis that leads to the malignancy, that leads to the cancer. So you want to stop all of this from happening. Okay. Last question. Which of the following regions within an androgen receptor protein contains zinc finger motifs? A, B, C, D, E. Which one will it be? B, yeah. The domain that binds to the DNA needs to have zinc finger motifs to be able to bind to them. And that, no, there's still more. One more question. A steroid-responsive tumor cell has abnormally increased activity of MAP kinase signaling in response to the steroid. Which of the following aspects of steroid signaling may be involved in this abnormally sustained signaling? So the steroid is leading to MAP kinase activity. Which one of them? Membrane-initiated steroid signaling. Yep. MAP kinase is associated with membrane-initiated steroid signaling. Correct. And I think that is it. That is all the questions from the chapter, the entire chapter. The unit, not even the chapter, it's a full unit. We did the full unit here. We did what we, what happened here? We did from the book. I hope it helped, and inshallah the recording will be uploaded and everything. Yeah, we did these three pages 160 through 185 even. If anybody has any, unfortunately, I will not be able to do any more PAL sessions for MOL. I don't think, because I'm very behind on my studying, and I will be free like next week. But you have your exam next week. I want to do the PAL sessions next week. Yeah, if anybody has any questions about other lectures, feel free to ask me whenever you want. They can give you my number. I'm pretty good still with MOL. I've been studying for my step 1, so I'm still decent with MOL. I think I can help people. And yeah, thank you for joining, and I hope you the best of luck on your renal exam in 2 days and your MOL exam next week. Also your English tomorrow probably.